Welcome to The Explainer. Today, we're diving deep into one of biology's greatest detective stories. We're going to crack the code on how scientists actually read the secret language of proteins, the very building blocks of life.
Okay, so let's just jump right in. We all know proteins are essential, right? They're these tiny molecular machines that basically run our bodies. Everything from digesting your lunch to fighting off a cold. And what makes a protein do its specific job is its secret code, the precise, unique sequence of its amino acid building blocks. Figuring out that code is the key to understanding, well, everything. But how on earth do you read something you can't even see?
You know, the best way to think about this whole process is like solving a giant invisible puzzle. Figuring out this sequence, what scientists call the protein's primary structure, is a bit like trying to reassemble a document that's been put through a shredder. Your job is to figure out what each tiny strip of paper says and then piece it all back together in the right order.
And the stakes here are incredibly high. The order has to be absolutely perfect. A single amino acid out of place, in a chain that could be hundreds or even thousands of units long, that can be the difference between a perfectly healthy cell and a serious disease. So the goal here is crystal clear. Uncover the exact order, from amino acid number 1 all the way to the very end.
So let's rewind the clock a bit and look at the original detective work. I mean, long before we had supercomputers and high-tech labs, scientists came up with this incredibly clever, really methodical way to solve the puzzle, one single piece at a time.
The strategy is just pure logic. It's beautiful. First, you do a quick analysis to get a parts list of all the amino acids in your protein. Then, here comes the clever bit. You use chemical scissors to chop up that long protein chain into smaller, more manageable fragments. But here's the trick. You do it again on a new sample with a different pair of scissors that cuts in different places. Once you've figured out the sequence of all those little pieces, you just have to find where they overlap to piece the whole thing back together.
And these aren't just any old scissors, they are incredibly specific. For instance, an enzyme called trypsin will only cut the protein chain right after a lysine or an arginine. Another chemical, cyanogen bromide, will only cut after a methionine. It's this amazing precision that actually makes the entire strategy possible.
So, after we use trypsin, this is what we get, our first set of clues. We have 4 separate fragments. Now, we can figure out the sequence of each little piece, no problem, but we have no idea what the overall order is. Is T1 at the beginning? Is T3 in the middle? Right now, they're just disconnected puzzle pieces on the table. This is where the magic happens. We take a fresh batch of our protein and use our second chemical, cyanogen bromide. This gives us a completely different set of fragments. And these new pieces are the absolute key. Because they were cut in different spots, they're going to create overlaps with our first set of clues.
And here it is, the aha moment. When you lay out both sets of fragments, you can start to see how they connect. See? You might find that the end of one fragment from the first set perfectly matches the beginning of a fragment from the second set. They create these little bridges. By lining up all these overlaps, the one and only correct sequence for the entire protein just reveals itself. It's brilliant. Puzzle solved. Okay, so that was the classic old school way. Now, let's fast forward to the modern era and see how this detective work got a massive upgrade. Technology has given us tools that have completely revolutionized the game, with unbelievable speed and precision. I mean, the leap forward is just staggering. The classic method was genius, for sure, but it was also painfully slow and you needed a lot of your protein to do it. Modern methods? They can spit out a sequence in minutes from a sample so tiny you literally couldn't see it. One of the absolute game changers here is a tool called the mass spectrometer. The simplest way to think of it is as an impossibly sensitive molecular scale. It measures the mass of a protein or its fragments with such incredible precision that it can tell the difference between individual amino acids. And it gets even cooler with a technique called tandem mass spectroscopy. It's pretty wild. Inside the machine, a single peptide fragment gets isolated, and then it gets smashed into smaller bits in something called a collision cell. Then a second spectrometer measures the mass of all those new little pieces. And since every amino acid has its own unique mass, scientists can just work backward, calculating the mass difference from one piece to the next to read the sequence.
But hang on a second. What if there's an even bigger shortcut? What if we could just skip analyzing the finished protein entirely and go straight to the source, the original blueprint written in our DNA?
And yeah, we can totally do that now. Sequencing a gene from DNA is incredibly fast, and we can use that to predict the amino acid sequence. But, and this is a really critical point, the DNA doesn't tell you the whole story. After proteins are built, they often get chemically modified. Special bonds might form that are crucial for its final shape, and the DNA blueprint won't show you any of that. That's why scientists use both techniques together. They're complementary, and you really need both to get the complete picture. So, we've been deep in the details here, but why does all this matter so much? Well, it's because these amazing techniques allow us to zoom out, to go from solving a single, tiny puzzle to mapping the entire library of proteins that makes an organism, well, tick.
And that brings us to this huge concept, the proteome. This isn't just about one protein anymore. The proteome is the entire set of proteins, all the thousands of different types that are active in a cell or an organism at any given moment. It's the whole cast of characters.
And that is why this is so profoundly important. Mapping the proteome is at the very heart of modern medicine. It lets us see the intricate machinery of life in action. It shows us what's supposed to happen in a healthy state, what goes wrong in disease, and it gives us clues about how we might be able to fix it. So think about it. We've gone from painstakingly piecing together one protein over months to mapping thousands of them almost instantly. We have this unprecedented ability to read and understand the fundamental language of life itself. So the question is no longer how we read the blueprint. The real question is, what should we build with it?